In this video, we are going to be discussing the last few topics in Chapter 17. First, we are going to go over what happened to the pyruvates that were created in the process of glycolysis. Now, one of the things that I should specify is that glycolysis itself is going to happen in the presence of oxygen or in the absence of oxygen. Now, the cell itself have to look at what conditions am I at. If the cell has oxygen, then what's going to happen is that those pyruvates are going to be processed through aerobic oxidation and oxidative phosphorylation to overall being converted to 6 molecules of CO2, 6 molecules of H2O, and as you can see, we are going to be producing energy in the form of ATP, which I've highlighted in yellow. If the cell does not have oxygen, then depending on the type of cell that we're going to be talking about, in the absence of oxygen, so this is, yes, we have O2. We have no O2. Then those pyruvates are going to be either processed through anaerobic glycolysis, and in the case for yeast, then they can those pyruvates can be converted through anaerobic alcoholic fermentation into CO2 and ethanol. Now, anaerobic glycolysis takes those pyruvates and makes them into lactate. Let's look at those reactions more precisely. So in section 17.4 in your textbook, we are going to first discuss what happens to those pyruvates if they're processed through anaerobic metabolism specifically. Remember, anaerobic means that we do not have O2 present in the molecule. So in that case, understand that pyruvate is going to be converted through the enzyme lactate dehydrogenase into the molecule of lactate. This reaction, as you can see, is an oxidation reduction reaction in which understand that your NADH is oxidized to NAD plus and your pyruvate is going to be reduced to lactase. Okay? Understand that the NAD plus can be used back in step 6 in glycolysis in order to generate that NADH, the 2 molecules that we generate in step 6. Understand that this reaction is actually exergonic and it is catalyzed, as I mentioned previously, by the enzyme lactate dehydrogenase. So understand that in organisms like yeast, anaerobic conditions, again, we don't have molecular oxygen around, so it's in the absence of oxygen, we are going to be converting pyruvate specifically to ethanol. So this happens in 2 steps. In step 1, that pyruvate will be converted to acetaldehyde and CO2 with the use of the enzyme pyruvate decarboxylase. Then on the second step of the reaction, understand that that acetaldehyde is converted to ethanol by the enzyme alcohol dehydrogenase. Okay? So, if we look overall at this process, what we're looking is at the conversion of pyruvate through ethanol. So understand that during this process, we have NADH being oxidized to NAD plus. That specifically happens when we are converting acetaldehyde to ethanol. Because remember, aldehydes get reduced to primary alcohols. So that's why we have our coenzyme being oxidized, and all of that is catalyzed by the reaction called alcohol dehydrogenase. As I mentioned previously, pyruvate, if we have oxygen, that means that we're under aerobic conditions. Those two pyruvates are then going to be converted into a brand new molecule that is called acetyl-CoA. Acetyl-CoA specifically is what enters the citric acid cycle. Citric acid cycle has several names, which are also known as the TCA cycle. That stands for tricarboxylic acid cycle, or Krebs cycle. All of those names for the cycle are synonyms, but understand that the details of how pyruvate gets converted to acetyl-CoA and then enters the citric acid cycle are going to be described in detail in Chapter 19.